All right, everyone, welcome back, or welcome for the first time. So today we're going to be doing... Okay, all right, I guess we're live streaming now, hopefully. So welcome back, or welcome. So today we're going to do microarray analysis. So this is kind of putting the cart before the horse a little bit, but given that we did RNA-seq analysis, dedicated the last few meetups to doing RNA-seq analysis. However, hopefully today you'll see that performing microarray analysis is much simpler than doing RNA-seq analysis, and we should be able to do everything within the hour. And we'll begin by showing you how to use this tool called Geo2R, which is a tool made by NCBI that allows you to easily analyze publicly available microarray data. Unfortunately, it's not sophisticated enough to do automated analysis of RNA-seq data, but maybe that might be something in the future. So again, to begin, either if you have a dedicated project for this meetup, that's great. Please open up that project. If not, just open up RStudio. And as always, we're going to install the package from GitHub. And so that's remote : install _ GitHub Duke CCT/cbrg. And then we need to specify this force parameter to be equal to true. And again, the reason we do that is to override any existing cbrg package that you happen to have installed. So obviously for me, this works without any problem. So hopefully this is the same for all of you. And once you've installed it, or maybe even reinstalled it, just load your package by doing library cbrg. And again, it's okay to do all of this in the console now, because this is code that can be discarded once you run it. And finally, hopefully there's no error with this. We can just do launch project, and this is meetup number 7, so we do launch project 7. And hopefully after all of that, you will see an R Markdown file that corresponds to today's analysis, and it's called How to Perform Microarray Analysis. One final step, if you're going to annotate this, which I strongly recommend you do, make sure to save as. So you want to navigate to your, oops, let me see. You want to navigate to the folder that is that corresponds to your project, and you just want to save it there. And if you've done it correctly, you'll see it appear in this files pane here. All right, so again, as always, we have to install a few packages this time. Fortunately, unlike for DE-seq and RNA-seq analysis and all the other things that we did in the past few meetups, there are just 2 more packages to install today, and that's GeoQuery, which is what we will use to download data sets from Geo. So I should say Geo is Gene Expression Omnibus, and that's where you find this Geo2R tool. And this package called LIMA. And LIMA was originally made to perform differential expression analysis on microarray data. That's what one of the I think the MA stands for microarray. But LIMA has since been used for all kinds of omics analysis. And we'll go into why this is such a powerful package and a little bit on the stats part. So, and again, all of this super clean code was written by Paul. And all you have to do is click play. And obviously, again, for me, this was trouble-free because I already have these packages installed. Hopefully, they'll be trouble-free for you also. So these are 2 really nice, well, all this little script here looks to see if you have these packages installed. If you don't, it will install the packages, and then it will load them all in one nice chunk. All right. So hopefully, all of that is good. So for today, the data set we'll be taking a look at corresponds to this paper here, Identification of Potential Caloric Restriction Mimetics by Microarray Profiling. I have a particular interest in caloric restriction. Lots of my research has had to do with caloric restriction, and so I thought this would be an interesting, and I think this is of kind of general interest, especially to people at the DMPI. The one downside to this is I wasn't paying too much attention to when exactly this was published, so this is a 16-year-old study, but hopefully it was done well, and we can get something useful out of this. So just a brief description of the study. Their objective was to perform gene expression profiling of liver tissues from mice that had been exposed to long-term caloric restriction. So beginning, I think, at 5 months of age until for 20 months or something like that, very long term. The additional goal was to see, and their point was, okay, lifelong caloric restriction was associated with lots of these physiological benefits, which we know, but the downside of it is it's lifelong caloric restriction. So adhering to something this extreme is difficult to do. So they were interested in seeing if they could identify any so-called caloric restriction mimetics. So they tested a bunch of different drugs, one of which is metformin, and they also looked to see if late in life caloric restriction that was only, in this case, I think 2 months long, could recapitulate the effects of lifelong caloric restriction. Sadly, it can't. That's the take-home message. Of the different interventions they tried, metformin ended up being the closest. Short-term, this 2-month-long consumption of metformin was the closest, had the closest effects compared to lifelong caloric restriction. So this is a rich data set, and after today's session, if you'd like to look at other comparisons and compare your results with the results of the paper, then there's a lot that you can use here to practice your skills. So before we begin with all of this, let's actually just use the GEO2R tool. So each kind of project on Geo is associated with an accession number. So this one paper, the data set associated with this corresponds to this GSE 2431. So I'm just going to copy that so that I can remember what that is. And we can just go to Google and type in, I think it's just Geo and Geo datasets. And there are literally thousands of datasets in this repository. And I think you can just type in things like caloric restriction, and you can search for datasets yourself. I think that's how I found this in the first place. Or if you have this accession number, then that makes it easier. And I believe it's just this one. So this page gives you all kinds of useful information on this particular study, including the abstract and information on author, submission date. It also has these few things here. So just briefly, so this is some of the terminology that Geo uses. So one thing is this thing called a platform, this GPL. And platform corresponds to, if this is in the case of microarray, it corresponds to the type of chip that was used. So in this case, it was an AFI chip, U74A. And if it was sequencing, it would tell you what sequencer it used. And then it gives you information on these samples. So each sample, it has these first 3 letters in common, GSM, and then some number. And then next to it, it tells you what the sample ID, what group it corresponds to. So there's control and the different interventions, long-term caloric restriction, metformin, and a couple of others. And then finally, if you want to bypass all of this, if you just wanted to download the raw data yourself, you can just do it here. So actually, the GeoQuery tool we'll be using today, we'll be downloading normalized data, but that's already been pre-processed. But if you wanted to just download the actual raw data, that's possible too. So you can just click this Analyze with GEO2R, and it'll take you to the GEO2R tool. And you can see here, or you could just directly go to that tool and put in the Geo accession number here. And it shows you all the samples. In this case, there are lots. And so today we'll take a look at control and long term. So to do that, so in the case of, we'll do a simple analysis. We'll just compare two groups today. You first click on define groups, and you give the groups names. So I'm just going to call one group 0 and call the other group 1. So 0 is my reference group, 1 is my the one I'm comparing to the reference. So to assign samples to a group, you can just click on them, and then click on the group ID. So now I've assigned all the controls to group 0. And if I scroll down to long term, so that's an n of 4 for each group, I assign all of these to 1. Right. So once you've defined the groups, you scroll all the way down, and you just click Analyze. And this obviously will take a few seconds to run. Right. And then it gives you all kinds of useful information. So there are these various diagnostic plots. So for example, this one shows you the distribution of expression across the samples. Since these are normalized, you would expect them to be comparable. So that way you can actually do statistical comparison that's valid. Then shows you distribution in another form. There's a nice volcano plot. There's an MA plot. And then there's this thing called UMAP, which is kind of like a non-linear principal component analysis. And it shows you that just straight away you can tell that the for control and the for long term, there's nice separation of these two groups. So that's nice to see. There's a Venn diagram. And importantly, there's a table of differentially expressed genes. By default, the Geo2R tool gives you just the top 250 most differentially expressed genes. And it has like log fold change, gene symbol, and so on. So that's it. That's kind of all there is for at least a simple pairwise group comparison using the Geo2R tool. And a couple of other, oh yeah, and then you can set various options. So these are all the defaults, and you can change them depending on what you want to do. There's a profile graph. Not really sure what this one is, but importantly, there's this R script associated with these changes that you've made. And if you change any of these options, or you change any of the group definitions, that will be reflected in this R script. So this is a really nice tool, both for having reproducible analysis and also for educational purposes. So this is really comprehensive. Like there's code on everything that you see here. So today we'll just look at the differential expression analysis and maybe a little bit at some of the visualization. So what we did was, if we go back to, copy pasted much of this code, but added a few changes here and there. And those, any differences have been highlighted in the code itself. So before I begin with this, does anyone have any question about Geo2R or microarrays or anything? Cool. All right. So I won't be going back and forth between this too much, but I just want to take a look at, show you the first part, so that you're loading these relevant packages. Today we won't be running UMAP, so we just have GeoQuery and LIMA, and these first few lines show you how to actually load the data. So. And you can see like these lines here look like these lines here. So the very first thing we do once we've loaded our packages is the first function that we use is this thing called GetGeo. It's a nicely named function. And it takes in the relevant accession number. And then you specify this. So this GSE matrix equals true. I'm not actually sure why they even specified that here, because that's the default parameter, but I guess they were being clear about it. And what this means is if you set this to true, you get the expression data, normalized expression data in the form of a matrix. So this is the so-called series matrix. And then they have this additional parameter here, this annote GPL equals true. And these different platforms, so in this case, the array chip, they have annotations that are associated with them. And if you set this annote GPL equal to true, I think these annotations are frequently updated. So that update is reflected if you set it to true. And then you can, if you run all of this and you just assign it to an object called, you can call it whatever you want. Obviously, I'm just calling it what Geo called it, a GSAT. You can see, let me just move this. You can see up here that GSAT is a large list. It's quite a big object. And if you look at your console, you'll see if the download happened as it was supposed to happen, and it looks okay. So I put a little question mark geo here just to show you, this is really annoying, just to show you what this function is and what the various parameters mean. So that's just if you're curious about other parameters that are in this function. Just reduce the size. So let's try to see what this GSAT object is. So one way is to just use this class function to see what kind of object it is. And R tells us that it's just a list, which we can also confirm that it is in fact a list. And actually, if we click on it, we see that you can see it's quite a complex object. Now, this one line here, sometimes a given experiment can be associated with multiple platforms. So in this case, maybe it's like multiple array chips that they used for whatever reason, because like some chips can be used to do differential splicing analysis, others can be used to do just gene expression analysis. So this line of code here on 86 looks to see how many such platforms there are, and if there's just one, it'll just assign one to this object called IDX. If it's more than one, then more numbers will get assigned to that. So let's just run that as is. And just to confirm, if we look at all of what line 86 did, if we go to the console and just type in IDX, you see it's just simply the number 1. So we use that IDX, which in this case is just 1, to subset this GSAT object using the familiar double square brackets. So if we just run this like this and override the GSAT object, we now see that it's, if you notice here, it's changed from a large list to a large so-called expression object. We can confirm that on line 89 by, again, rerunning class. And we see instead of getting a list, we get this thing called an expression set. So an expression set is very similar to the summarized experiment object we discussed in the context of RNA-seq analysis. And again, if you click on it, you see that it is once again this so-called S4 object, which is a really powerful and convenient way of putting together lots of disparate information in a coordinated, efficient way. The downside to it is this contains this various sources of information, so navigating this can be a little tricky. And there's a lot of useful information, like obviously the gene expression values and phenotype data and things like that. But there's also a lot of random information, not random, but like not so useful for the user, like what was the person who uploaded, what was their email address, things like that. And so I've just, we've noted here that this GSE followed by some numbers, so that's the series accession number, and it can be associated with multiple platforms. So that's what this one line is trying to see. And then if we look at the class, we see that it's an expression set. All right, so the expression set, there are different, just like with the summarized experiment object, there are different ways to access information from within this object. There are some really handy accessor functions which make life easy. Or you can, as I'll show you later on, you can kind of use R's built-in at symbol to extract information that's relevant. So one thing that you can look at is you can use this fvar labels function. So again, I mean, the downside to all of this is this new expression set object, there are a new set of functions to learn, but fortunately there aren't that many. And once you run this kind of analysis a few times, you'll see the same functions keep popping up. So for example, we see that there are all these sorts of information associated with this particular expression set. And so I put a question mark fbar labels, and that'll tell you what it does. So I'm just going to skip that. There's this function called, so one thing to notice with these names is they are not particularly programming friendly, by which I mean, for example, some of these names have gaps in them, and that's always not a good thing. So what you can do is you can use this built-in make.names function, and that'll kind of like clean up these names. And importantly, it will also match these names with this so-called top table format. And top table is something that we will encounter towards the end of our differential expression analysis. So this is just a step that is included in the GEO2R code. So if we just run that, and we run fbar labels again, we see that now instead of these gaps, we have dots. So it's just a very small thing that this line of code does. So we can take a look at what samples our GSAT contains, and we can just do that using this call names function on GSAT. We can see that these have all these various GSMs. Again, it's somewhat useful. You know how many samples you have, but you don't know what each of these samples, which group it corresponds to. So I put this line here. This is not in the GEO2R code, and I bet there's an easier way of trying to find out what the identities of these different GSMs samples are, but this is one not very readable way of doing it. And essentially what you're doing here is this expression set object consists of multiple so-called slots. And the @ symbol gives you access to these slots. So you can see that GSAT contains one big slot called phenodata. Within phenodata is another slot called data. And within data, there's this title object. So I don't just happen to know that. I just, like if I want to see what's going on, like you click on the various slots to try to see what that logic is like. But like I said, this is just kind of sort of like an FYI thing. It's not particularly, this information here, you can just as easily get it from GEO2R itself. If you scroll up to the samples table, you can see that the first 4 objects are con and so on. So this is just a programmatic way of getting the same information. All right, so that's that. Now this part here, I'll do my best to explain this properly. This part here can maybe get a little bit tricky, especially because we're not dealing with the data frame, we're dealing with this expression set object. So let's try to go through this line by line. This line here, this 112, and the subsequent few lines, you can kind of think of this as the programmatic or the coding equivalent of clicking on the relevant samples. So just as we clicked on the different samples and we assigned them to either group 0 or group 1, these lines of code are doing the same thing. So the first thing is to create an object that is just this string that has zeros in the location of the group 0 samples, ones in the location of the group 1 samples. So that since the first 4 samples are control, the first 4 values are 0. And then I can't count what number this is, but whatever number that is index position, those 4 consecutive ones are the long-term caloric restriction. And everything else is x'd out. So everything else becomes an x. So if we just run this, we see, just to confirm, if we run GSMS in the console, we see that it's just this string. Now this line, the next line on line 113, what it does is it takes this string and it splits it into individual characters to create a list. That's what this str split equals is doing. So if we just run that, we get SML. If we run it, we see we have a list of these individual characters. I wanted to show you one thing, just to show you why we need this double square bracket 1 indexing thing. It's something that I hope to one day actually get right the first time, but this is one of those things in R that I try something, so I'll try this. And you see that if you just run this str split without this double square brackets 1, you almost get the same thing, but it's, someone explained this nicely. This is kind of like a train car. So the train, like a train is made up of multiple cars. So we can think of this as one car, and within this car, there are passengers. So if we just run this, we get the car with the passengers. We're not interested in the car, we're only interested in the passengers. So to see or to gain access to the passengers within the car, we do this extra step here. So if we rerun this and do square 1, so now it's like the passengers have left the car. I don't know, that kind of helps me, but I still keep making this mistake. So the point is, at the end, you want SML to be a list of characters, which it is. Then the next thing to do is, it's line 116. We do this which SML is not equal to X. So let's run that and see, and we assign it to an object called SEL. Let's run it to see what we've done. So what this does is it's asking what is the index of the non-X samples. So it's another way of saying that is asking what are the indices of the samples that are either 0 or 1. Right? And so we know, so it'll return 1, 2, 3, 4 because those are samples group 0 controls, and then 17, 18, 19, 20. And these are these ones here, the long-term caloric restriction samples. What we can do is we can use this list of indices to extract the zeros and ones from this SML object. Now this one seems a little silly, so let's just run this to see what this looks like. So now if we look at SML, we can see that we've gone from this list of characters of zeros, ones, and X's to just zeros and ones. And this is something that is not needed for these steps here, but we will need it later on, line 152. So if you want to go back and go through this code and kind of like move back and forth between this line and line 152 to make it clear why we have this step, that's probably the best thing to do. And then finally, what we have is this GSAT. We can use this SEL, just again reminding you what that is. So these are just numbers. And in R, just to remind you, if you want to select columns, you can use this square brackets notation with a comma in it. And so what this means is, from this GSAT object, I want all the rows and comma, and then the columns that I want, I need to put the numbers of those columns here. So in this case, if I run this, I get rid of all the columns that correspond to the X's. So now I just have the columns corresponding to group 0 and group 1. So let me first just run this call names again. Actually, no. Let me run this long line again to show you that the GSAT object still has all the original samples. But when I run line 118 and I rerun this, now GSAT just contains 4 controls and the 4 long-term. So all of this is like the clicking and assigning to the 2 groups that we did using the Geo2R tool. Okay, so now we have a GSAT object that contains the samples of interest. The next step is, I mentioned that these, when you run this GetGeo to get the series matrix file, these are all data that have been pre-processed, by which I mean the data have been normalized and background corrected. And this is something that's essential to do for microarray chips. And we do this so that we can validly compare samples with each other. One thing that is not necessarily done is Log2 transformation. Sometimes users do it, sometimes they don't. And we need to do Log2 transformation for a few reasons. One is it's, since we're using linear regression, we're assuming a normal distribution, and oftentimes gene expression data are not normally distributed, so one quick fix is to just log transform it. And also the LIMA package that we'll be using for the differential expression analysis requires Log2 transformed gene expression values. So what GEO does is, because for whatever reason it's not, I don't know why this information is not just included in like the metadata, but anyway, since GEO is not confident that the data have been log transformed, it has this line here. So that's these two lines 126 to 128. And there's a lot going on here, but broadly what it does is it's a little test to automatically detect if your data have been log transformed or not. So we'll go into the details of that in a second. So before we do that, let's just take a look at what the expression values are for our GSAT object. So to do that, we can use one of these, I mentioned these accessor functions for the expression set. So arguably the most useful one is this exprs, and that's the function that extracts the expression information from this GSAT object. So if we run that and assign it to this object called ex, we do that, and then we can view it, and we see that we have, and this is universal for bioconductor like containers, like these summarized experiments or expression sets and so on. The rows always correspond to the things that are being measured, so in this case, genes, and the columns correspond to the samples. So we can see we have 8 samples, 8 columns, and 12,500 genes. So there are lots of numbers here, but let's take a look at this first number, 12.9. GEO is now trying to determine if 12.9 is the raw 12.29 or if it's a log transformed 12.29. And to do that, it first creates a list of these quantiles going 0 to 1 for the expression object. So if we run that, and rather than looking at the details of these, what this function does, we just look at the output. It gives you the like 0th quantile, 99th quantile, and what numeric value that corresponds to in your expression set. So it's these values here. It then performs this logical operation, or the series of logical operations on lines 127 and 128 to ask the question, do these look like they are log2 values or raw values? So there are a bunch of different things going on here, but if you just look at the first one, this QX5, so it's indexing into this QX list. So 5 is 1, 2, 3, 4, 5. Is this one here? And it's asking, is this above 100? And it is. It's way above 100. So if it is above 100, this log C will be true, and if it's not, it'll be false. Now, and if this log C is true, if we skip down to line 132, all of this is saying, if this log C is true, I want you to log2 transform. Because the logic here is, if you see a value, your 99th quantile is 12,285, that's probably a raw expression value. Because if it were log transformed, the raw value would be 2 to the power of 12,000, which is unreasonably high. So that's kind of like the little test it does. So if we run that and take a look at log C, it's true. So it's like, this definitely is not log transformed. And this is some text to repeat that. And so if we run this line, so if log C, so this means if true, I want you to run this. So it does a couple of steps. One is it will look for all the values that are zeros. because obviously if you log transform 0, you won't get a sensible, it'll throw an error. So you specify in all those locations that you want, you're saying that it's not a number. So the log2 function will ignore those numbers. And for every other number, it'll just log2 transform. And we'll save this back in this GSAT object. So if we run this, there was an error. OK, just because of the way this code is formatted, go to before I, so the start of line 132, and then run it. So Command Enter. So now if we look at GSAT again, that 12.9 is now 3.6. So we can confirm that the log2 transformation has occurred. OK, so a couple of questions. Just in the interest of time, I'm just going to, so how would you compare the distributions across the samples? Can anyone think of a quick way of doing this? There's the, so if you look at line 139, that's a little bit of a clue, because that's saying that's a ggplot way, which unfortunately is not as quick. And then there's a much quicker base out way.
So you do a histogram.
Yeah, very close. I think you could just do hist. Let's try it. Is it just hist? Yeah. Hist. Have to do expression. This might or might not work, but so unfortunately, this is giving you a histogram of all the values. So if we instead do boxplot, and I don't have a good explanation for why boxplot doesn't do it. Boxplot gives you the distributions for each sample. That's interesting. I don't know why there's this kind of divergent behavior. It doesn't just give you one giant boxplot. Like, it's clever enough to know that you want the boxplots for the different samples. Who knows? So that's one quick way, and kind of as homework, try to do it with ggplot. It's a little bit unfair because there's one step that we haven't covered, but we can maybe cover it at the end if there's a little bit of time, or like next week. All right, so we have now we have selected, we have our samples of interest, and we have it log2 transformed, so everything is good. And we are ready to do the differential expression analysis. So I mentioned previously that, I've changed some of the lines. I mentioned up here this SML, like why do we do this? We need it at a later line, and it's not 152. I'm just going to say we need it later, because that line might keep changing. That's here. So we do so let's just run SML. So that's just a character vector. And to remind you that this is something we saw previously for the RNA-seq analysis, that the groups that you're comparing, they need to be in the form of a factor. And that's because you need to tell, R has to know, so factors have levels. So R has to know what the levels of your factor are to know what it's comparing to what. And just to further explain what that means, if we just do factor SML, we go from this character vector to a factor. And it looks very similar to the character vector, except now we have the 4 zeros, 4 ones, but we also have their levels. So this will tell the subsequent statistical models that 0 is our base group, our comparison group, and 1 is a group that we're comparing to 0. So that's important to do. So we just assign that factor to an object called GS. We can make names for the groups. So if we just do make.names, this will produce this. So the groups are just XO, X1. That's all there really is to it. And I guess the reason this step is done is because LIMA or GEO doesn't want these levels to be 0 and 1. It wants them to be XO and X1. So to do that, you can take this XO and X1 and assign them to the levels of GS. So we use this levels function here. So if we run this and take a look at GS again, now we see instead of zeros and ones, we have XOs and X1s. Again, I'm not really sure how big, I think this is really a formatting thing, but these are just, these steps are, these lines are included automatically in the GEO2R code, so I think it's just worth going through them because they're in there. Line 150 is, so if we look to see if there's something called group within GSAT, like there's nothing there, it's just a null. So what we can do is, this is where these expression set objects are really powerful. You can kind of on the fly just start adding information to them. So this GSAT object, we can add group information. So we do GSAT $group, and we assign this GS. So now when we run GSAT $group, we see that we've taken this GS group factor and we've kind of attached it to the GSAT object. And sorry, I think I'm trying to think of why we even need that step. I believe it's because when we make our design matrix on line 152, GSAT has to know, so it takes two things. Yes, no, that's it. Let me run this model.matrix first to show you what the output is, and then we'll go through it. So we create this model.matrix object, this design matrix. And what this design matrix is, this is similar to something that we went over in the first RNA-seq meetup, where we were talking about how we run linear regression or linear models in R. And so in this case, effectively what we're doing is we're just doing a bunch of t-tests, but we do it using this linear regression approach. And in this case, we're just comparing two groups, control with long-term caloric restriction. So in this case, our model will be a linear regression model, which will, we will have as many linear regression models as we have genes, because we're making all of these 12,000 pairwise comparisons. So the way to read this would be gene expression is equal to intercept beta naught or beta 0 plus beta 1 times either 0 or 1, depending on what you want. So this is known as a dummy variable. It's called dummy coding. So in the case of our control, control's a 0. So if we go through that equation, beta naught times, so basically beta naught plus 0 times beta 1, we just end up with the intercept term. So the intercept term is the mean expression value for our controls, for each gene. Conversely, for the long-term caloric restriction, there's a 1. So the equation for this half will become beta 0 plus beta 1, which is equal to the expression of the mean expression of the long-term caloric restriction group. And so we can further see this intercept here, this beta 1, as being the difference between the control and the caloric restriction group. And the statistical test is asking, is this difference significantly different from 0, which is the null hypothesis. Again, very kind of hand-wavy, probably best to kind of write it down and see it with pictures, but this was to some extent covered in the first RNA-seq meetup, so if you're interested in some of these details, please refer to that video. And if it's still not clear, which there's no reason, I mean, the good reason why it might not be clear, please email us. And the way we can construct that design matrix is to use this model.matrix function. And the model.matrix function requires two things. It requires the data set, so that's the GSAT, and then it requires this formula, which should be similar, should remind you of what we saw when we were running the DE-seq function for RNA-seq. So we have this familiar tilde, and then we have the name of the covariate. So in this case, it's just group. And the reason we do use linear models for any of these analyses is because if we were just doing this, then yeah, it's kind of a little bit silly. You're just doing a t-test. But what if you had batches? You want to do batch correction, or you have other, we haven't looked at it, but this data set might also contain information on other covariates like sex or age or things like that. So if you wanted to include other variables in your model formula, you can easily do it here. So it would be group plus, and if you had a batch term, you would just do group plus batch or something like that. All right, so we have a design matrix, and I wanted to say design matrices can get a little tricky. Even like this is the simplest case, but for more difficult, more complex cases, by definition, it becomes more complex. There's this series of videos, if you've never come across it, this is great to know, called StatQuest, which is just generally, and the person who runs it is a person named Josh Starmer, who actually, I think he used to work at UNC, so he lives in Chapel Hill. And it's just a series of great videos on statistical concepts and bioconductor. I think there's some stuff on bioconductor, R in general, and it's done in a really funny way with cute animations. So he has this series on design matrices, and then I have this, so we're about to start LIMA for a thorough description of, and then surprisingly, like oftentimes guides for especially these kind of heavy statistical concepts tend not to be very readable for people who aren't statisticians. I think one of the exceptions is the LIMA guide, which is really well done. So, and that also will go into more examples of much more complex comparisons and things like that. All right, so we have this design matrix that's here, and you can see that the column names are intercept and group X1. So this line on line 156 is just cleaning up the column names. So again, if we look at design after running that, we see instead of intercept and group X1, it's now X0 and X1. And that's just a cleaning up thing. And so now we can finally get to running LIMA. So there are a couple of steps, a few steps in LIMA. One is really simple. We just fit the linear model. And we use this lmfit function. And lmfit requires two inputs. It requires your data set, this GSAT, and the design matrix. And you will see that GSAT, there's correspondence between these row names of the design matrix and the column names of GSAT. And that's really important. If those two don't match, if there's some clash, you'll get an error. But having gone through all of this code will ensure that that is in fact the case. So what this is doing is it's just fitting a linear model for each gene. And that's it. And that's what the fit is. And it does it it's blazing fast. So we could kind of stop here because it's already done the linear regression for all the genes. But as we went over in the RNA-seq meetups, with omics data, or these kinds of data, the best, so in the case of RNA-seq, we were looking at log fold changes, and we went over the fact that the measured log fold change for a given comparison is actually not the best estimate of the log fold change, which sounds self-contradictory. It's actually a combination of a prior expectation of what that difference should be, what that fold change should be, and the measured fold change. So we saw this example of log fold change, so-called shrinkage, especially for low count genes. There's a very similar concept applied here, and that concept is called empirical Bayes. There's a very similar approach applied here, this empirical Bayes, but in this case, this line here, 161, what it's doing is it's applying this empirical Bayes approach to the standard errors associated with each gene, with each comparison. Why is this important? If you just run the linear regression naively, using the measured standard error, we use those standard errors to work out the test statistic, which in this case is the t-statistic. And of course, the higher the standard error, the smaller the t-statistic, and the less likely the chance of seeing significant difference in expression. So what this step here does is it moderates, the term is called moderation. It moderates these standard errors so that, especially for higher standard errors, they become smaller. And that's because what it's doing is it's working out some kind of average between, so it calculates the mean of all the variances in your entire gene set. So that's one value. And this empirical Bayes is, what it's doing is it's calculating some kind of average between that value and the measured variance. And if this part is not very clear, especially with my hand motions, there are two things. One is there's this excellent guide here that goes through this step by step. And there's this YouTube video by Raphael Irizarry, who's a huge name in genomics and is also a really good teacher. He goes through in even more detail, the actual maths behind it. And it's genuinely, it feels like, I said this to Paul, it feels like cheating still, but the actual maths behind it is not too much. Basically, the effect of this is it shrinks the standard errors to a prior mean or a prior expectation. And this, in effect, is a good thing because a lot of the, especially for when you have a low n number, this gives you a much more accurate estimate of the variances. And if we run that also happens really quickly. And this is additional parameter 0.01 that corresponds to what's known as the proportion. And it's just asking how many of these comparisons do you think will be statistically significant? I believe that's what the thing is. Again, I don't know why they put this here because that's the default value, I think. But that maths is a little beyond me. This part all of it is explained in the LIMA guide. And yeah, so we've done one fit, which is just fitting the linear model, then a second fit, which is applying this empirical Bayes approach. And finally, we can use this second fit, this fit2 object, to actually get our results table. And to do that, we use this top table function. And top table takes in fit2. We have to do some kind of multiple comparison adjustment. The default is FDR, is Benjamin E. Hochberg. And that's what the FDR corresponds to. But there are others available. There's this B statistic, and actually that kind of corresponds to this proportion value here. And that's the log odds of seeing differential expression. So if B is equal to 0, that means there's a 50-50 chance of seeing differential expression. I'm a little confused by this, but it's basically, if you don't have any missing values in your data set, the way that the top table ranks your genes, it'll be identical whether you use this B statistic or you use the p-values. And then this last bit here is different from the GEO code. GEO by default will set this number to 250, so it'll just give you the top 250 genes. If you want all the genes, you have to specify the number. And so that's equal to the number of genes. And to get that information, you just do the n row of your EX object that you created earlier. So if we run all of this, we get this TT object, and we can click on it. And you see that we have all kinds of information, some really useful, some maybe not so useful. There's a lot going on. So this final line here selects columns of interest. There might be other columns, but to start off with, I thought these were pretty important. And then you can arrange this in ascending adjusted p-value. So you start off with the most significant. And if we run all of this and look at TT again, we now see we have Affy ID, Affymetrix ID, the corresponding gene symbol, gene title, p-value, and adjusted p-value. And the top gene is, this is the real test. So it's this one here. Ferritin. I mean, something might have gone. I hope it's the same as the one we saw when we did this analysis here. No. I ran this previously and I got exactly the same output. So I'm a little concerned that I'm not seeing the same thing. I wonder why that is. Anyway, I'll work that out. and try to see if I can see why this is. But that's it. That's how to do differential expression analysis on publicly available microarray data. As you can see that there are lots of these like little fiddly steps here to get everything just right. Unfortunately, there's no way around that. But the good thing, at least what I find, is each of these individual steps, there's not much going on. So it's just like a little bit of cleaning here, cleaning there. And what's great about Geo2R is you can play around with these settings, like try out different samples, adjust different options, and you can see how it's reflected in the code. So this is a really good learning tool, and I wish there was something like this for everything else that we're doing. But yeah, that's kind of it. And obviously I've skipped all the stuff about like Venn diagram, volcano plot. The other slightly unfortunate thing is all of this is base R, so it's not as readable as Tidyverse. So with 3 minutes left, are there any questions?
So I guess I'm assuming like a lot of these little steps we talked about, you can just copy and paste.
Yeah.
Put your own data set. The other thing I had a question about was the section where you had the double bracket 1.
Yeah.
What did that one actually signify, and is that something you might have to change at some point, or is that kind of a constant?
So we saw that in a couple of places, like up here, that double one.
Yeah.
So so in this case, let's take a look. So let's just run this again and take a look at what we've made this SML object, and it looks fine. Oh wait, sorry. Let's remove it. Run it and take a look. SML. And so what we see here is, sorry, I have my Siri on. Go away. What we see here is it kind of looks like a character vector, but if you look closely, it's like 1 to 20 something. All of this is actually within something else. So if we just run this like this and try to run this, this proves that I don't really understand R. This worked. This subsetting worked. Oh, did it? No. So what it did was, so it didn't work. What it did was, so if we look at SML again, we see that again it's all of this contained within this one. So it's really, and actually we can. So it looks like there are 20 something objects in SML, but if we look at the size of SML itself, it's just SML just contains one thing. So when we do run the steps next, so that messes up this SEL thing. So if we run this line here and look at SEL, which is supposed to be the indices of all the non-X values, it gives us this 1, which is not right. So instead, if you do index into 1 like this, and take a look at SML again, you're kind of like going 1 level below to where you want to be.
I guess my question is it always a 1 though?
Does
that 1 actually signify something, or is it just?
It depends on your split parameter.
So this could be, so in case you can see, like there are actually 32 here. No, it's, you can have as many as you want. So you can kind of think of it, Paul, correct me if I'm wrong, I kind of think of it as multiple, again, slots. So in this case, we have a...
It depends on the split. If you split, for example, for 0 or whatever,
Yeah.
you will obtain a list of 2. If you split for another character, you will obtain a list of 1 or 3. Or whatever.
Oh, okay.
Then you have to select. So you are splitting by nothing.
Oh.
Obtaining just 1. But if you split, no, 0, and maybe X is a wrong example, but if you introduce a character in your vector,
Yeah.
like an A or whatever, and then on the, yeah.
Oh, got it.
And then split for A, yeah. Now you will obtain a list of 2.
Oh,
And if you are in the, or you should. Oh, so run GSMS again.
Oh.
You should deal with it.
It's interesting. Oh, no. For whatever reason, it split it, but it didn't split it into like 1 and 2.
Yeah.
It just put it in the first slot. It's this part like, this is why the tidyverse is so great, because it's just you just deal with data frames. I have to confess, like when it comes to like lists and vectors, I usually I try a bunch of things until something works, which is not a great thing to confess. But yeah, it's this kind of stuff it's a little bit arcane. But final thing, I know we're over time, but I came across this really great thing called Tidy Data Tutor, and you just put in code in here and you can run it. And so this is this data set called Palmer Penguins, and it just has information on, as the name suggests, penguins, and you can do all kinds of things. But importantly, it shows you at the end of each pipe what each line is doing. So it shows you what the select line is doing. You take your input and then you select these 2 columns. And if you go down, it shows you what group by does. Group by takes, in this case, species, and puts them into 3 groups. And then arrange tells you what it does, is it arranges according to bill length from highest to lowest. From highest to lowest. And slice cuts the first value of the 3 groups. This is really clever, yeah. And if you wanted to do something like, you can do summarize, mean length is equal to mean, bill length mm. If we run this, you see it's recognized that I now have an additional step and shows you how summarize works. So this is, I want to see what the limits of this thing are, but at least for these pretty simple but widely used functions, it works nicely. So that's it. Thank you for paying attention. And next week onwards, we'll be going through proteomics data and analysis. Thank you.